On est prêt. Je peux me chercher. Oui, s'il vous plaît. Ici, s'il vous plaît.
Good morning. Bonjour, everyone. So I'm happy to be today with you to present this session around artificial intelligence and bioinformatics. We have great researchers with us. Some are old, some are young, but however, even if they are young, they are doing a lot for the science in Tunisia right now. So in fact, we will start with this session with Professor AHMED REBAI. He's one of the eminent researchers here in Tunisia around bioinformatics. And now he's initiating in artificial intelligence in Tunisia. He's one of the founder of bioinformatics and biostatistics in Tunisia and even in Africa. He has more than 250. He has more than 250 publications. So today we will present around. Est-ce que vous pouvez enlever le micro, professeur Tabé? So Professor AHMED REBAI today will give a presentation around artificial intelligence and the new era of computational biology and knowledge-based minutes to present pre-work, and after that we will have 15 minutes, I think, around question and answers. Thanks.
So just I'm not hearing you very well, but I hope that you do. So may I start?
Yes, Professor, you can start.
Okay, thank you very much. First of all, I apologize for not being able to join you at the Institut Pasteur, and I thank you for the kind invitation to share with you with some of my, may I say, not knowledge, but probably wisdom that I have get in 30 years of scientific career. And this is why I will be talking more about ideas than about technical issues. And the choice of this title for the conference is that we are living now a revolution of AI, which is going in every scientific field, and particularly in the analysis of genomics, or more generally, omics data, which are the raw material for what we call computational biology or bioinformatics. So what new knowledge this implementation or this introduction of AI in computational biology will bring us? So we have some kind like doing what we have now, and what can we expect from what we're having now, and the development of the future will bring to the human knowledge. So before starting, let me express in few words my solidarity with Palestinian people and in their stand against occupation and what they are living now from killing children and women and so on. So my greatest solidarity with the people of Palestine. So the first three natural questions we may ask when we are talking about AI, computational biology, and knowledge is what we mean by these words. The answer might be very clear, but I think it's always good to ask ourselves what are the meaning of the words or the concepts we are using. So let's start by what is computational biology. we can define it very roughly as any use of computational, statistical, mathematical methods, algorithms, programs, and so on to analyze biological data. But in this case, this is an all-inclusive definition. That means we can put it in it everything that uses one of the methods in analysis of biological data. And for myself, I see it a good definition. So when we talk about theoretical biology, which comes back to the 1960s, about statistical genetics, which goes back to the 1920s with Fisher work, biostatistics, bioinformatics, which is more recent, can trace back to the 1980s, systems biology at the end of the millennia, the previous millennia. So for myself, I really don't care about definition. And I do think that clear-cut definitions are just good for making journal ranking, to building communities. And at the end, it will result in building boundaries between different science disciplines. And I believe, and I do believe, strongly believe in transdisciplinary science. When we have the hybrids that can emerge from the marriage between different sciences, this is where good things happen. So just going very quickly, computational biology, bioinformatics, whatever, we are dealing with the analysis of biological data, whatever their kind. It can be omics data or any kind of data. This is still under the umbrella of computational biology. AI, of course, it's a huge, immense technology landscape. If we are, for example, doing computational biology, much of the work is inside the red circle you have here. That means we are doing machine learning, deep learning, etc. But again, computational biology, when we are generating data that are analyzed by computational biology, we are using many other technologies in that landscape. Brief history of artificial intelligence, good to remind that it comes back, we can trace it back to the 1950s with the first paper by Alan Turing. And remember that these papers of artificial intelligence were published in journals of psychology and philosophy. That means that we have a strong background in cognitive science where we are talking about artificial intelligence. And we can trace back really the foundation act of the artificial intelligence to the Dartmouth Conference in 1956 by these guys. And one of these guys is Claude Shannon. And Claude Shannon, you know, is the father of what we call information theory. And he was the first one to actually imagine an experiment with artificial intelligence. That's, remember, the mouse in the maze that he called it Theseus, which is a Greek god. Theseus. This was an experiment using technology that are currently in use in robots, just to implement in one of the first experiments of artificial intelligence. Now, the question, what is scientific knowledge? What we mean by knowledge? We know. Knowledge more widely, it's very big, it's philosophical concept. You can write many books, and actually many books have been written on what we call knowledge, but let's focus on what we call scientific knowledge. It's a collection of reliable new information about the physical world. And the humans, all the living beings, are part of the physical world. Of course, we are not talking about angels, demons, gods, and so on. We're talking about the physical world that we can see, that we can study, that we can examine. So how we gain knowledge about that physical world? By intensive processing of data, collection, experimentation, and of course, analysis. So the scientific knowledge is the body of laws and theories. Laws are observed patterns of phenomena. Theories are more wider. That means that we have a systematic explanation of the phenomena. The roots of scientific knowledge is good, again, to remember that the evolution of the scientific thinking in humans, and we can trace it back maybe the first traits, you can trace them back to the Greeks, probably before, but because we know that Greeks have taken knowledge from the different civilization, from the Persian civilization, from the Chinese, from the Africans, Egyptian, of course. But what we have written in history is that we have, for example, we know the books of Aristotle, we know the elements of Euclid, and these are 3 centuries or 4 centuries before Christ, and these are the act of the birth of modern science, what we can call modern science. That means that we have the basic of logic, the basic of induction reasoning, the basics of axiomatic deductive methods that have been founded in Greece. The scientific revolution, really what we call the modern science, if you want, like something like very clear, we have clearer methods, we can trace it back to the mid 15th century with the work of Vesalius on the human body. The same year, Copernicus have published the heliocentric model, which is a revolution against the Aristotle theory of the geocentric model. That means that all planets are moving around the Earth, and Earth is the center of the universe. You're saying, no, it's the sun, at the center of the universe. At that time, of course, the universe was the solar system. But later, 50, maybe something like 30 years after that, we have one person, which is a Danish scientist, but I think this is my opinion, because I think that he was the founder of what we call today the big data, because he was in a philosophy of collecting data on stars, observation. And at that time, he said, science is not collecting observation. It's collecting observation data. But to make science, you need to process this data. And process, that means you clean the data, you analyze the data, you interpret the results. That means this is actually what every scientist today is doing. And Tycho Brahe was also concerned by developing new tools. Of course, if you observe, you see the limits at what you have today in like equipment, instrument technology. So we're just trying to develop more and more precise, more accurate instruments to do your study. This is what happened for sequencing, for example. Sequencing, if you come back to the 1970s, you have Sanger sequencing, and then next generation sequencing, and now 3rd generation sequencing, and so on. So we are again developing more and more accurate technology in sequencing and beyond in the computational biology field. So if we summarize, we can find actually on different documents, different papers, this kind of division of the scientific knowledge. The first period, that what we can call empirical science, like making experiments, collecting data without really analyzing them and deducing some laws or theories. The second part, which can be traced back again to the 15th century or 16th century, is the theoretical part. This part that goes from 16th century to mid 20th century, when we'll do a lot of theories, mainly in physics, but also in biology. Remember that statistical genetics, or what we call biometrics or biostatistics, have done a lot in this first part of the first half of the 20th century. And then we have a new paradigm with artificial intelligence, the paper of Alan Turing, 1950, which is what we call the computational science. We have computers. We can do a lot with computers. It changes really the way of doing science when we can make computations, calculations very easily, very quickly. And accumulation of this power of computing have resulted in a tipping point that happened maybe in the 2000, with the change of the millennia, and we moved strongly to what we call the data-driven science, with artificial intelligence, with statistical learning, with etc., with data mining, and so on. So the dates are quite indicative. Some of the publications show it like that. 1,000 years ago, we are doing almost only empirical science. 100 years ago, we are doing theoretical science and then computational science. And then now we are in the data-driven science with what we have in machine learning and artificial intelligence. So if you want to put that knowledge, scientific knowledge, it's important to talk about that, to explain where we are going in the next years. So if we want to draw a tree of scientific knowledge, and the tree, of course, in the tree, you see that you have roots, so on, you have branches. So I just asked ChatGPT to draw me a tree of scientific knowledge, just putting a question in ChatGPT. So it gave me something like that. It's very artistic, actually, but it's have no sense for me. See a lot of formula, a lot of drawings, and so on. So okay, it's good art, but it's not informative at all. So let's go back to Aristotle. Aristotle have already drawn what he called the tree of knowledge. And the tree of knowledge at that time, of course, we are 4th centuries before Christ. He has philosophy, of course, we have technology, they call art and technology and scientific knowledge on the other page. But if we want to draw a tree similar to that one today, and this is what I tried to do, very roughly, of course, because we have details, we have different intrication between branches and so on. We can say that before the 15th century, we were mainly doing empirical science. We moved to theoretical science, what we can call hypothetico-deductive approach. That means we are putting hypotheses, we are making experiment, generating data, and then we are verifying or not the hypothesis, and then we are concluding about some theory. Data analytics, Tycho Brahe, remember I said that he was accumulating a lot of data, saying that we need to analyze the data, and then the theoretical science emerged. We have able to put the data into equations, mainly in physics. And then comes the era of computational science, which is based, remember, on inductive reasoning, particularly when we're talking about statistics. And I have a statistical background more than a bioinformatics background. And statistics, we are making inductions. That means we are moving from the special case of the data we have to generalize to the theoretical population we think that sample or that data represent. So this is the tipping point of artificial intelligence and the millennia, and I invite every one of you, mainly the young people in the assistance, to read this seminal paper, this very important paper. And of course, you can read the comments on that paper. Many comments have been written on that paper, but it was a paper by Leo Bremen, who is the father of what we call decision trees or random forest and so on. And he said that actually we can have two different types of views. We have the statistical world or the statistical culture, which is based on models, very interpretable models, and we have the culture of machine learning, more general artificial intelligence. Sorry?
Professor? We have some problem of connection. Can you please shut down your camera, if it's possible?
Sorry?
On a des problèmes de connexion. Donc, est-il possible, s'il vous plaît, de fermer la caméra? La vidéo.
Donc,
عندنا مشاكل نتاع كونيكسيون سي احمد، دونك كان تسكر الفيديو سا سري بيان.
سكرت
الكاميرا
c'est bon.
Non, ça marche.
Très bien. Donc, je viens de fermer ma caméra.
امشي l'icône فيها caméra.
Oui, c'est bon, j'ai appuyé dessus alors. Oh.
وزينا نشوفوا فيه.
Elle est encore... Alors, attendez.
Je me dis ça.
Alors, je vais peut-être... Désolé, je vais arrêter... Stop cam, voilà.
Là, ça marche. Merci.
Le postage est bon?
Non, on ne voit plus vos slides.
D'accord, désolé, je vais partager de nouveau. C'est bon maintenant?
Oui.
Okay, so let me continue. I'm sorry for these technical problems. So I was saying that we are starting from the new millennium, we are moving from theoretical computational science to data-driven science strongly and strongly. So actually the heart of the conference, this was an introduction, quite a long introduction, but it's very important to put these things in the good way. So the question I will address in this conference particularly are how advanced computational biology and artificial intelligence can improve our scientific knowledge in life sciences in general, but in particularly in genomics and the omics family. And the second question is that, very linked to this one, is that it is currently possible, just a quick overview that we are doing today, and what might be possible in the near future or in the future in 1 century or 2 centuries. So I will start by the second question. So you might ask yourself, why am I starting with the second while I can have put it as a first question? So this is just to, sorry, to give you time to think about the first question when I am talking about the second one. So the second one is what we are doing, what is currently possible in the future. And this, I will just focus on one thing, very extremely informative in that sense. 2 years ago, Japanese team have launched what they call it the Grand Challenge of the Nobel Turing Challenge, NTCI, Nobel Turing Challenge Initiative. This is launched by a university in Japan. And the challenge says that, can an artificial intelligence system completely automated, that means without a human intervention, make Nobel level discoveries, that means can make scientific discoveries that are at the level of getting a Nobel Prize. So it might seem quite strange, but actually it's not that strange if you look how humans are making science, or how artificial intelligence system can make science. We see a huge difference. We as humans, we are trying, while we're making scientific discoveries, we are trying to maximize our chance to make significant discovery. That means discovery that have impact, that have sense for us, for our knowledge of the physical world, but also how we can act on that physical world. So this is what we can call a value-driven approach. So we want an added value of the scientific discoveries we are making. AI scientists, that does not exist today, but can exist at any time. What they will do is that trying to maximize the probability that making a discovery of any level of significance, just discovery after discovery after discovery, and it doesn't make sense for them to, if that discovery had an added value or not. So this is what we call it an unbiased exploration of hypotheses, or exploration-driven approach of science. So if you want to get an analogy between the intuition-driven design of experiments and exhaustive generation of experiments and biased exhaustive generation of experiments, this is actually what we are doing mostly in omics approaches. For example, we are sequencing the whole genome, we're sequencing the whole exome just to look for a mutation that is responsible for a disease. We are sequencing all the RNAs in a cell just to see which is overexpressed and underexpressed, which regulates what, and so on. And we can do that because we have the technology to do sequencing, because we have powerful computing facilities to make the analysis of this data. So human scientists, AI scientists have different approaches, different philosophies in addressing science. And remember that we have an example in human scientists that have using almost the AI approach of science. That means we generate exhaustive experiments and we see from that exhaustive experiments what's come out. And this is the 2012 Nobel Prize on the discovery that mature cells can be reprogrammed to become pluripotent. And the experiments that have been done just extracting genes and making leave one out experiments, that means just knocking out one gene and see the impact of that another gene and so on. So in that sense, I can deduce which gene is a key gene in reprogramming the cells. So will such an approach, which is based on AI only, like exhaustive exploration of the hypothesis of the universe of possible hypotheses we can have, is efficient or not? We have the experience of omics. We are almost doing that. We're generating a lot of data, and from these data, we are exploring a number of hypotheses. To be possible, this unbiased hypothesis generation and verification should have three components. You should have an hypothesis generation system. You can have a series of machine learning and reasoning systems that analyze your data and extract information understandable by a human from this data. You should have a robotic-based experimental system. That means no human intervention in all this. The machine learning system hypothesis ask the robots to make experiments, get back the data, and then make the conclusions. This is a brute force approach, actually. So like we are destroying everything. We are just generating hypotheses and testing them without knowing which one is very likely to be true. Will it be efficient? I just remember you that in the first time of artificial intelligence, most people thought this is not a good approach. Science is not that, not generating a lot, all the hypotheses and testing them one by one. But actually, the evolution of AI have shown the inverse. It have shown that we can do it very efficiently and get very successful results. And just remember, just the papers in the last few years by DeepMind, Google DeepMind, and the field of games, a chess game, for example, on Go game. They have generating systems that do not know the rules of the game, but they are able to learn from playing, and they were much more efficient than all the previous systems that have been based on either computational science or artificial intelligence. So this approach of exhaustive exploration of hypotheses is very likely fruitful and very likely to be successful in the near future. Just to show you this, the series of papers in Nature published on the chess game. But games and science are quite different. We know that in games we have almost a finite space of possibilities. In science, it's an open-ended and very likely infinite world or universe of hypotheses or possible scientific discoveries. In science, we have description that is well defined and is not accurate. That means that science is built on, but I mean, what Popper, the philosopher Popper said, falsifiability. That means you have a result that is now true, and after that, that is demonstrated to the limits of that result are shown, and we have a new theory or new knowledge, like in mechanical, the Newton mechanics and the Einstein mechanics, different schools. And also in science, evaluating hypothesis is very costly and time consuming. That means if we want to explore a large number of hypotheses, it costs money, but it costs time also. However, despite these limits, I do think that in the few coming years, maybe half a century or a century, very likely I will not be there with you to know when it will happen. But I do think that it will happen. If we solve all these issues, and they are solvable, they are manageable with new technology, with new computational facilities, and this will transform the way we are doing science. That means that we will get access to a wider space of scientific knowledge that is impossible to access with our human capacities. Now, just to show you this figure, where you see that this is the space in green, the space of scientific knowledge that we means humans plus AI or AI alone, we can know. The limits are not clearly defined. What we actually know today is this red dot. What is knowable, we can discover as a humans only, with a human mind, with a human facilities, human reasoning, is that pink circle. What we can know with humans combined with AI facilities, that means a human-centric AI hybrid systems, is that blue circle. And all the rest, all the green, it's very likely, very large, that with a human capacities we cannot do it, but we can rely on artificial intelligence to make an exhaustive exploration of this universe of hypotheses. So remember this figure. I will show you something similar with the alpha missense system of Google DeepMind in annotation of non-synonymous variants in genome sequences. Again, if you want to see how it works, this system, this artificial intelligence-based system, how it can work to generate hypotheses, to verify them automatically, you just imagine that we have an artificial intelligence system that generates exhaustive experiments. From that experiments, we have different data sets. For example, here, example, we have 3 data sets. Data set 1, when we analyze it, we can be inspired to get a set of hypothesis A. Data sets 2 and 3, they inspire a set of hypothesis set B. These 2 sets of experiments, we generate new experiments of hypotheses. We generate new experiments to verify if these hypotheses are true or not. If they are, we have them verify it, and then both of them can inspire another set of hypothesis C. So we are exploring the universe of hypotheses, of scientific hypotheses that can bring new knowledge to us. So now I'm moving. So this is the future. This is the second question. The first question, sorry, that we are doing now, and we can be doing in the near future, I chose here very few, it's not exhaustive, just an examples of the challenges in computational biology that can be solved by AI, with the help of AI. And we have two examples here in red that have already been solved by the Google DeepMind, again, with their AlphaFold system for predicting precise structure of large molecules of proteins, particularly, and the quick analysis of polymorphic genetic variation. This is the alpha missense system that have been published just 1 month ago in Science. I will show you the paper. They just annotated all the possible variants that result in a change of an amino acid in the protein sequence. This is the AlphaFold. You have it if you go to the MBL EBI site. You can just enter protein name or gene name, and you get the structure as done by the AlphaFold system. And the AlphaFold system is based on AI. It's not like doing that like we did in protein structure modeling, like having a template and homology and then trying to do energy minimization and so on. So they're just learning from that what we know about structure of other proteins from their primary sequence alone. So just we have proteins with primary sequence. We have their structures experimentally resolved, and the AlphaFold learned from that to get the prediction of the structure from the primary sequence of the protein. So this is the paper 2 years ago. And if it's quite, I mean, funny if you read the advertisement about these products, because these are products of Google DeepMind, they are commercial products. How they are presenting this thing. This is why biologists are turning to AI. The ability to predict protein shape computationally from its genetic code alone, that means from primary sequence, will help dramatically accelerate research. We are here in an AI-based system. That means you are exploring, I mean, exhaustively the hypothesis. And this is an example of what you can get with the AlphaFold. You have a structure that is learned from the primary sequence. Of course, we have learning database with primary sequence and structures resolved, and you get a probability of the different regions the system is predicting the structure. Means what is in dark blue, let's say that this part of the structure is very reliable. You can rely on it, while other parts might be not that reliable. This is the alpha missense, second example, released 1 month ago, of what we can do with AI in the field of genomics or omics more generally. We have humans till now, I mean, in maybe 20 years since the first release of the human genome, we haven't identified a lot of SNPs, single nucleotide polymorphisms. We have annotated some of them. We have a clinical impact of some of them. The all we have resolved is 2 million. Theoretically, there is 71 million non-synonymous SNPs that can be present in the human genome. Non-synonymous or missense, that means you have a change of an amino acid in a protein sequence. These are variants in the genomic sequence that result in a change of an amino acid. 71 million possible theoretically. Only 2 million of them, or over 2 million, have been already found. And the system, the alpha missense system, is using learning with AI. Not using the classical approach. Of course, it is combining the structure, the impact on the structure of the change of the amino acids, using their AlphaFold system, okay, which is based itself on AI, and they are making multiple sequence alignment to come out with a probabilistic classifier saying if this SNP or this missense variation results in something that is damaging or that is benign, or can have uncertain effect. This is the paper in Science, very interesting. You can read it, I mean, in your by yourself and just see how artificial intelligence alone is really implemented in analyzing the genome sequences or missense SNPs. And just to show you here, among the 71 million possible missense variants, almost 90% have been classified either as likely pathogenic or likely benign, and 11% are uncertain. Again, alpha missense makes better in terms of precision, accuracy than all the predictions already in use, prediction systems based on classical approaches of bioinformatics. So this circle reminds you the first circle that I show you in exhaustive generation of hypotheses in science. This is exhaustive generation of missense variant, because most of that variants have never been seen. You see here the variants, the 69 million variants, non-synonymous variants possible that have not been found already in the human genomes, but might be found in the near future, have been examined. What we know, what we are confirmed by human experts, represent only 0.1% of these 71 million SNPs. What we have already seen in human genomes, in the sequences that have been sequenced worldwide, is only 6% of the 71 million possible missense variations in the genome. And this is a summary. We have, you see here, 71 millions possible missense SNPs. We have 69 millions absent, never been observed in humans till now. We have 5,000 have been found in one individual. We have 2.7 millions that are very rare variants that are actually mutation, and we have 70,000 that are common variants with frequency more what we can call common than 0.1%. Sorry. So I'm coming up to the end of my time, but I'll be very quick on this third part of my talk. So the first part, the objective of the first slide, to show you that with artificial intelligence, we are not just empowering computational biology or the omics sciences. We are changing the face or the way we are doing omics and bioinformatics, in the sense that we will have access to systems that can not only analyze the data and help us get meaningful results from that, but also generating hypotheses by themselves, automatically generating and testing hypotheses. And this is the future that I do believe will happen in the few coming decades. So another issue that we have in artificial intelligence is what we call the black box issue, that when we are making machine learning, actually using approaches that are very advanced, like deep learning and so on, we don't know exactly what happens inside the box of the learning algorithms, which features are important, which are not. We know that the system is learning something from the data and giving an output, something meaningful to us, predictions that can be used. But how can we know more about what happens in that black box? And people are talking about what they call explainable artificial intelligence. So when we are handling large-scale biomedical data, including omics data, of course, for a patient, you have a patient, you want to do personalized medicine. So you have at the same time the clinical records, the genomics records of that individual. We have a lot of other features, and you have a model that have been learned on large number of individuals, and you're coming out with a prediction for that individual, saying that you very likely have a cancer of that type. So what happens is that if you use a system like black box model like neural networks, deep learning, or even some methods that are more simple than the deep learning, you are high in accuracy. You know that the model you are learning is very accurate, but it's very low in interpretability. It means how you interpret that prediction. And on the other side, you have less precise, less accurate methods, but are highly interpretable. And this is the word of statistics, actually. Yeah? We have here statistics. We have here the two cultures, remember, the statistical culture and the machine learning culture. So what we can do if a system here have learned data, it's a nice model, I know it's very accurate, and that model give me that a patient, a woman here coming to a doctor, she has 70% of having a breast cancer of that type. So the doctor is just saying, you have 70% of getting a cancer. So the lady is asking, can you explain why I have that cancer? Or what is the impact actually of having this kind of cancer? Can it be cured? Should I take drugs? Should I make maybe preventive measures and enough? So the question is how we make this prediction more interpretable, understandable for a person. We are here in a very sensitive environment. We are talking to humans. We are not talking to robots, talking to humans. So we have to take into account fairness, I mean, accuracy issues, avoid any bias that can happen from the system because the system is learning from data. So how much bias is there in the data that have been used to learn the machine learning system or to learn the artificial intelligence system that is making predictions. So in the pipeline, we have a lot of possible bias. So what it can be done, and actually is being done today, is making this black box model more transparent, more interpretable by using what we call rule extractors, symbolic reasoners, and this is again made by artificial intelligence. And so that we have more than saying you have 70% probability of having a breast cancer of that type, than something meaningful to an individual. That means your chance or your of having a cancer is 70% because you have in your blood a biomarker called TP53, which is a protein coding gene, which have been already associated to cancer. And there is many studies, 500 studies, for example, have been proven that people who have that biomarker have cancer. So, just talking human to human to explain in simple terms how you come out with that prediction. So at the end, this will result of systems like we have data, the real world, data that are collected by doctors. So people are coming to the doctor for asking for help if they have health issues. We have the genomics, the image, the clinical records data that all are used by black box to learn a black box model, deep learning typically. We come out with a prediction system, but before providing the prediction to the doctor to advise the lady coming, we pass through what we call an explanation AI system that gives sense to the prediction you have coming. It's something like a second round of analysis of the data to come out with simple rules that are understandable by humans. So at the end, let me conclude, but about what I think about AI, artificial intelligence, and its application particularly in the omics world, is that it's not just a tool to improve or accelerate our ability for scientific discoveries. More and more, it will be implementing a new scientific revolution, a new paradigm in science, which is based on completely automated system to make scientific discoveries, from generating hypotheses, from analyzing data, to verification, generating verification experiments, making them, collecting the data again, and moving to until the discovery of making new discoveries in science. So I will end my talk with my great thanks to you for your invitation, but also for your listening. And if you have question, of course, I will try to answer. I'm not sure to have all answers to all questions. This is the way scientists work, human scientists at least, but I will be more than happy to listen to your questions. Hello? Hello? I'm not hearing you.
Can you hear me now?
Now I'm hearing you.
Okay, interesting. So I just said that it was a very nice presentation. So AI is helping like scientists or researcher in different fields by streamlining the drug discovery, like reducing costs for developing safe and effective treatment. But it started like in the 15th, but still have, I think that still have like policies and ethical challenges. So my first question is can you say that the AI is still in its infancy? And my second question is about when we talk about AI, we talk about data, and we know that data are really very diverse and very heterogeneous. So how AI scientists deal with the bias introduced by the diversity of these data? Thank you.
Okay, well, the sound was not very clear, but I think I captured the question. Is that how we deal with heterogeneity in data and how can we correct for the bias it can bring into our learning systems?
Yeah, that's it.
Yeah, this is one of the challenges actually in the artificial intelligence or machine learning communities, and more methods are developed to unbias the problems of, for example, overrepresentation of some data in the learning data set. For example, if you are learning something in personalized medicine, and if in your data set you have mainly people or data from European descent individuals, so it will be automatically biased. Because if you use it, for example, for African people, when you have a very scarce data on in genomics on African genomes. So there are methods being developed to deal with that. Actually, we can find some papers in the literature, but this is one of the challenges that is being addressed in current research.
Thank you, Professor Bay, for this very insightful presentation that brings the philosophy part on how we do need to think about AI and its application in anything, but in science, let's say in large space, but also in genomics. So my question is more about, do you really think whenever we will be leading or developing AI tools that are supposed to conduct hypothesis drawing, then doing the analysis and conducting the very classical way of analyzing the data in order to come up with novel discoveries in science, to which extent the bias that you have been talking about that can be introduced through the data and the limited knowledge that now exists, whether it is the human knowledge or the knowledge that is covered with the physical data that is really existing, and that these AI scientists, as you called, will be really able to explore the fuzzy part on your on the figure you showed. So to which extent you think that this is kind of feasible in a times scale that we will witness, let's say?
Yeah, this is actually one of my strongest message from the conference. And if I look the way artificial intelligence have evolved during the last 50 or 70 years, and the developments that have been made at several levels, not only algorithms, also to technology, linked to robots that are making experiments. Now you have more and more automated systems for DNA extraction or sequencing and so on. So what is missing is that to have a system in between saying that, making the link between the system that analyzes the data and then suggest an hypothesis to a system that will make the experiment and generate new data. So I do think that will happen. That will happen. The time scale, I'm not sure about that. Actually, if I was sure, I would be like a fortune teller and I'm much richer than I am today. But I think that it will happen in a time scale like something like 100 years. Of course, when you are talking about bias in hypothesis exploration, actually the system is doing exhaustive exploration of hypotheses. So actually it will be the most unbiased possible system. So just seeing all the hypotheses without taking which data are more represented in the data sets currently available and if they can cause bias. I'm just exploring the whole thing. How much we can go quickly in exploring this exhaustively, this hypothesis space is uncertain, of course, but I do think that it will happen.
Yeah, hopefully we will witness that then. Thank you.
Welcome.
I have a question, Professor Bay, if you don't mind, everyone. So my question is about the fact that behind artificial intelligence, there is human. So when you are talking about hypotheses and the modeling part, there is human behind that. So my question is, if we want to lead in artificial intelligence here in Tunisia, what we should work on? Is it we should work on hypotheses? We should work on what kind of complexity we need to have for the data, or is it about modeling behind artificial intelligence?
Yeah, I mean, your question is in the context of Tunisia, what we can do to improve our abilities to get better expertise in artificial intelligence, and mainly if we apply that to medical or genomics data. That's the question?
Yes, it is.
Okay, thank you. Yeah, I do think that the good thing with artificial intelligence is, of course, as you said, it is a product or result of a human intelligence. But it will come sometime that it will be completely independent of the human intelligence. And you see a lot of films in the science fiction films saying that the robots can become, at one time, they can become autonomous. But this time, again, I think it will happen, but maybe in a larger time scale than I have said for the system generic hypothesis. In Tunisia, we have, of course, to develop our skills in the use of the algorithmic part of artificial intelligence, like our skills in making models from data, our ability also to generate this data, particularly in the genomics field. I mean, it's quite strange that in the whole Tunisia, we do not have till now a sequencing machine to make whole genome sequences, human whole genome sequences, or if any, it is not used already to do that. So we have a lot to do to develop our capacities in generation of data and also in widening the community to make the community become big of people mastering machine learning, data analysis using artificial intelligence tools to get more capacities in entering this new world of scientific knowledge.
Thank you. Thanks, Professor. So now we will move to the next session, which is all about oral communication. So I give the word to Hussein.
سيد احمد صباح الخير سيدي.
مرحبا. مرحبا حسام. يعني مش نخرج مش نخلي لك
In the meanwhile, just we'll have a presentation now, but in the meanwhile, maybe I have a last question. So I invite Slim for the first presentation. And in the meanwhile, I have one question for you. I really like the idea about the automatic generation of hypotheses. And we think about ourselves as rational beings, but in fact, our thoughts are very contaminated by the cognitive biases. So I think also that biology has failed in the recent years. Social sciences also has failed miserably, more than biology. So my question for you, with all this, the machine learning, the AI and everything, and this mega machine that will generate ideas and hypotheses, what is the role of a scientist in the future?
I don't think that I have an answer to that, but I think I do think that the humans will always find a way to give sense to their existence on Earth. So this is rather a philosophical question that I think that always when we're talking about artificial intelligence, it is the end of humanity, transhumanity. These are concepts that are being discussed in philosophy and social sciences. I do think that humans are the smartest guys on this planet, at least. Maybe other planets, it's another issue. But we'll find a way to give sense to their existence.
Okay, let's hope for the best. Okay. Okay. Thank you, Sid Ahmed. I was really great journey between the past and the future.
Yeah, thank you to all of them.
It's always a pleasure.
Young guys. Yeah, you are the future, right? Goodbye.
Thank you, Professor. Okay. حبينا نوريهم الجونس ما هذيك قلت لك نقلب البوسي اي اتس اوكي
في الزرب معناها
مشي راجل مشي terminé. نشوف